Hello, I am Professor Sims. In this video, we will be discussing tools for characterizing DNA, RNA and proteins, genetic engineering, and gene therapy. For Lesson 10, the learning objectives are we're going to be looking at different types of mutations that occur in DNA. We'll talk about some tests that are used to detect carcinogens, including the Ames test. We'll identify some tools that are used in genetics that were actually derived from microorganisms, so that's pretty cool. We'll look at how to use gel electrophoresis to separate DNA fragments and to visualize them. We'll talk just briefly about other techniques that are used in molecular genetics, like restriction fragment length polymorphism, RFLP analysis, southern blots, northern blots. Then we'll spend a little bit more time talking about PCR, polymerase chain reaction, and we'll talk about Sanger sequencing, DNA sequencing via the Sanger method. And then just very briefly, we'll talk at the end about some things having to do with genetic engineering and gene therapy. So let's talk about mutations. Mutations are changes in the DNA which are heritable, and they arise from a change in the amino acid sequence, and usually, it says sometimes, but usually the changes in the function of a protein, because structure equals function in a protein. A point mutation, that's a mutation at a single base pair, so it's very small, one base pair. There are effects of this, which you probably wouldn't think like changing one base pair could change all of that much, especially when we're talking about genomes that have thousands and thousands of nucleic acids. But no, one single base pair being altered can cause a silent mutation. So this is when the new codon actually codes for the same amino acid. So it's silent. It's not a big deal. But it can cause a missense mutation where the new codon encodes for a different amino acid. So now all of a sudden you have a different amino acid that's changing the structure of a protein. It can make the protein non-functional, okay? And then a nonsense mutation. This is where the new codon becomes a stop codon. So this is even a bigger deal because it stops translation. No more amino acids are being formed and the protein is not being formed. Missense mutations, they can retain the function. It does depend on the chemistry of the new amino acid and its location in the protein. So it can change the structure and the function of the protein, but maybe, just maybe, it doesn't change it. It depends on where it is. It depends on how different it is. It depends on a lot of things. But nonsense mutations, they produce what they're calling truncated and frequently non-functional proteins. So yeah, that's like what I was saying, is the amino acid production stops. So the protein is not being made, it's not being fully formed, and usually its function is lost. So this is just an illustration of your different types of point mutations. So a silent mutation has no effect. We had an A that was turned into a T. Well, GTT codes for val, and so does GTA. So no harm, no foul. This C was turned into an A. So instead of getting this amino acid, we're getting a completely different one. And then nonsense, this G was supposed to be a C. So instead of this amino acid being formed, and then presumably the next one, right? It's a stop codon instead. And then of course you can have insertion or deletion, which results in a frame shift. A frame shift mutation results from insertion or deletion of a number of nucleotides, but it can't be a multiple of 3, okay, because the codons are in multiples of 3. In order to shift the reading frame, it has to be something other than 3. So when the reading frame shifts, it alters every amino acid that is being produced after that shift. So it is a very big change in the amino acid production, which means that the protein that comes from that is going to be totally altered in its structure, and it's not going to function anymore. Spontaneous mutations happen during DNA replication, and then induced mutations, those are induced, they occur after they have been exposed to some kind of a mutagen, usually some kind of a chemical mutagen. These are either base analogs analogs or chemicals that modify existing bases, and chemical mutagens are usually introduced during DNA replication. Mutagenicity, talking about mutants, becomes important when you're looking at microbiology and you're looking at cell division and things like that, because mutagenicity is an indicator of carcinogenic potential, in other words, cancer. Okay? Auxotrophs are mutations that are specifically loss of function mutations, and these are associated with carcinogenic cells, mutagenic, cancer-causing cells. And you can detect auxotrophs by doing what is known as replica plating or performing an Ames test. So replica plating is where you compare the growth on a complete plate, like a bacterial lawn, and that is growing on a plate that has all of the essential required nutrients, versus the lack of growth that occurs on a media that doesn't have all of the nutrients. So I'm going to explain that a little more in the figure coming up. The Ames test is testing for a carcinogenic chemical compound, and I'll show you that some more in just a second. So this here is the replica plating. So over here you have media that has histidine. So this one is specifically looking for a histidine auxotroph. You have bacterial cells that are growing on a plate containing this compound that you're looking for. And they use this sterile velvet just to lift the cells up off of that plate. And they put it on media that contains histidine, and then they put it on media that does not contain histidine. Okay, and then they're going to compare those two plates to each other. What they're looking for are these mutants that can grow on the media that contain the histidine, but they do not grow on the media that lacks it. So it's just a direct comparison where they're looking for these bacteria that have mutated in such a way that they can live on this media containing histidine. This is the Ames test. This is used to identify potentially carcinogenic chemicals. So this one is showing a Salmonella histidine auxotroph that's used as the test strain exposed to a potential mutagen carcinogen. The number of reversion mutants capable of growing in the absence of supplied histidine is counted and compared with the number of natural reversion mutants that arise in the absence. Okay, so they add this extract from a rat liver that has a Salmonella strain that requires histidine to grow. So this is what's growing out on this plate. And then in this one, they have this possible mutagen. This is the thing they're testing. So in order to test to see if this mutagen is a histidine auxotroph, in other words, a possible carcinogen, they compare the growth of these two. So this is kind of the control, and then this is the test. And if they have a lot of growth on this plate, then that means that possible mutagen is in fact an auxotroph and can probably most likely cause cancer. So that's good to know. 12.1 is talking about microbes and the tools of genetic engineering. So we call this whole idea of genetic engineering and taking advantage of systems that exist in biology to benefit humankind. It's called biotechnology. So in order to be able to alter the genomes and to do things like recombinant DNA technology, this is all genetic engineering. Molecular cloning is the methods that we use to construct recombinant DNA and then also to get it to replicate in host organisms. And some of the tools that we use for this are things like restriction enzymes. Restriction enzymes are kind of like scissors. They're used to cut DNA in very specific places. And then ligation is like the glue that pastes DNA back together. And then, of course, introduction of recombinant DNA into host organisms is the last step of this. So this is figure 12.2. It's talking about recombinant DNA as artificial recombination of DNA from two organisms. In this example, it's the human insulin gene that's been inserted into a bacterial plasmid. And this recombinant plasmid can then be used to transform bacteria which gain the ability to produce insulin. So this here is very near and dear to my heart because my daughter, has type 1 diabetes, and she has to take synthetic insulin just every day for the rest of her life. And this is the process, this recombinant DNA process is how they synthesize the insulin in the lab. It's just amazing that we can do this kind of stuff. This is figure 12.5. Showing the steps in molecular cloning via bacterial transformation. Essentially, you have foreign DNA in a plasmid with an ampicillin-resistant gene. They're cut with a restriction enzyme, and then the restriction site, it's just showing that it occurs in the middle of this lac gene in a plasmid. So cutting this gene prevents the production of this specific enzyme called beta-galactosidase. The restriction enzyme leaves a sticky end on the foreign DNA, and this allows the foreign DNA to be inserted into the plasma because the sticky ends stick together. Adding the ligase reattaches the DNA backbones, and these are the recombinant plasmids. So you have foreign DNA, that's this in the red, and then the plasmid that you're introducing it to, that's this guy. Pieces of this foreign DNA essentially are being added to this plasmid, and then it reattaches, so you have a complete plasmid again. And then these plasmids are introduced into a growing bacterial culture, and then you have some bacteria that will take up the plasmid and then reproduce, so they'll replicate the plasmid. Some of them don't take up the plasmid. And you can actually visualize which ones do and which ones don't. Generally, the plasmid will contain some kind of a marker, like a flag or some kind of a molecular marker that makes it change color. And you can compare plates, like you can see on the plates which ones have the plasmid, the recombinant plasmid, and which ones don't. This one is a blue-white screening, and you can actually see the transformed bacteria. In this case, they're using reporter genes, and instead of seeing blue or white colonies, you actually have colonies on your plate where if you shine a UV light onto them, they'll glow because they contain these green fluorescent markers in them. Genomic libraries and cDNA libraries, these are really similar, and again, we're getting really heavy into the molecular stuff, so just know that we are just scratching the surface of this. I just want you to know what they are and how they are similar and how they're different. Genomic libraries, what they do is they clone fragments from one organism into a plasmid vector or into a bacteriophage. Remember, a bacteriophage is a virus that infects bacteria. cDNA libraries, they are using mRNA molecules, and they're looking more at genes that are expressed. So genomic libraries have to do with the gene being present. cDNA libraries have to do with genes that are being expressed. And then just down here, there are several different methods of transfection, like just physical methods that these things are performed with. You've got electroporation, gene guns, microinjection, shuttle vectors, and viral vectors. So the next two figures are just comparing genomic libraries to cDNA libraries. A genomic library, you've got DNA fragments that are recombined with plasmids, and then the bacteria that have the transformed plasmids are then grown out and replicated. And you can actually quantify these. There's lots of different methods. Then the cDNA library has to do with reverse transcription, and it's basically getting DNA, or what is known as cDNA, complementary DNA, starting with mRNA. So you're kind of going backwards in the process, which is why they call it reverse transcription. But then you end up with transformed plasmids, and again, those are grown out. Gel electrophoresis, agarose gel electrophoresis, is the way that you can separate different fragments of DNA, which is useful if you want to compare DNA samples from different groups or different individuals. Restriction fragment length polymorphism is where you can visualize distinct variants of sequences, and it's because they've been cut at different restriction sites. Southern blots are used to find a particular known DNA sequence, whereas a northern blot is looking at whether or not something is expressed, so that you're dealing with mRNA sequences. And a microarray is where you can compare and contrast samples. You can look at whether a gene is present or if a gene is being expressed. and you can compare samples, like just thousands and thousands of samples all at once. It's known as a high throughput method. Very, very cool. So this is gel electrophoresis. Essentially, you're pouring liquid agar or agarose into a chamber, and there's the little comb here. You let that solidify, and then you pull the comb out, and that creates these little wells. You load your DNA sample into these wells, and then you subject the whole thing to voltage. You want to make sure that the negative charge is close to where you put the DNA, and the positive charge is on the opposite end, because DNA does have an overall negative charge, and you want the DNA to migrate away from where you put it, and it does migrate based on the size of the fragment. So the smaller fragments will travel farther down the gel than the larger fragments. And then this lane here, this one has actually been loaded with what is called a DNA ladder. It's also called a DNA marker. And what that is it's DNA from a known sample that has been cut with different restriction enzymes in order to make DNA fragments of known sizes. And then you can use that. You run it along with your unknown DNA. You run it at the same time on the same gel so that you can compare the DNA that you're testing or investigating with that of known sizes. You can see, like, this band here is about 1,000 base pairs, whereas this one here is probably closer to like 300 base pairs. And that's what you use the marker ladder for. This is RFLP analysis, and again, it's looking at different mutations and different regions, different sequences in DNA, and again, you can separate them out using gel electrophoresis. They separate by size. This one is comparing the normal strand to a mutated, and you can see there's a band here that isn't here, and then this band is missing over here. So you can see that there has been some mutation. Polyacrylamide gel is very similar to agarose gel, but it's generally used to separate proteins instead of DNA. And then the protein it's been pre-treated with SDS. This is all stuff that is kind of outside of the scope of our class, but it's just examples of different ways that you can actually visualize these things. PCR is a way to, it basically mimics DNA replication that happens in yourself, and it's a way to amplify DNA sequences so that you can use them for other things, for experiments and things. In vivo is DNA replication, the stuff we did in lesson 9. In vitro is the artificial process, that is polymerase chain reaction. And there's different types of PCR. There's your regular PCR, and then there's also RT-PCR and qRT-PCR. RT-PCR is where you're looking at mRNA expression. So the RT is reverse transcriptase. qRT-PCR is where you can actually watch the amplification in process in real time. So the RT and qRT-PCR stands for real time, and the Q stands for quantification, quantify. The difference here really is that you can test for whether genes are expressed in RT-PCR, and in qRT-PCR you can see how much of a gene is present. In Sanger sequencing, you can actually discern the actual nucleotide sequence, like the A, T, C's, and G's. You can tell how many of them are in a DNA sample and what order they're in. And this is revolutionary. It's a huge deal. But I'm going to tell you, it is kind of old school nowadays, because now we have next generation sequencing, we have pyro sequencing, they're doing CRISPR now, really cool stuff. Essentially, you start out with your regular double-stranded DNA. That DNA is heated to, it's generally a temperature between 90 and 94 degrees Celsius. That detail is not important, but it is important to know that it's heated enough to where it breaks the hydrogen bonds that hold together the nucleotides, but it's not so hot that it breaks the phosphodiester bonds that hold together the backbone, okay? Then primers are added, and these are synthetic primers that are made in a lab, okay? These are not your RNA primers like in DNA replication. These are synthesized primers. Your nucleotides are actually also added to the reaction. Okay, so these are, in DNA replication, the nucleotides are already in the cell, they're in the cytoplasm. But in the lab, of course, you have to add it to the reaction. And then you also have to add a polymerase. This DNA polymerase is going to be a DNA polymerase. Usually it is TAC polymerase, TAQ. TAQ stands for Thermus aquaticus, which is a species of bacteria that lives in hot springs. Essentially, that works very similarly to the way the DNA replication works in vivo, except that it is happening in a test tube, in a lab, in a machine that cycles through these temperatures over and over and over and over again. And at the end of every cycle, you have an exponential increase of this DNA that has been amplified. Sanger sequencing really kind of capitalizes on the process of polymerase chain reaction. It's essentially amplifying DNA in vitro in the lab, except what they have done is instead of using your regular deoxynucleotides, right? They use those, but they also it introduces these things called dideoxynucleotides. The dideoxynucleotides, they have a little alteration here. So a hydroxyl group has been removed, and in its place they put a little molecule that fluoresces under UV light. And this is really useful because, so this is figure 1223. What they do is just like in PCR, they put in a test tube, they put the DNA template that they want to sequence. Okay, so we're sequencing this time, not just amplifying. PCR just amplifies, it just gives you more DNA. But if you want to know the sequence of the nucleotides contained within that DNA, you have to do something a little different. So in Sanger sequencing, they put in the test tube, right? They put the DNA, they put synthesized primers, right? Just like in PCR. They put the deoxynucleotides, just like in PCR. They put the polymerase, the enzyme, again, just like in PCR. But they also add these dideoxynucleotides. And what happens is you have all of the ingredients, you put it in a thermocycler, it cycles through just like it does in PCR. But what happens is that randomly, instead of a regular deoxynucleotide being added, you'll have one of these kind of funky, altered nucleotides that adds instead. And because the nucleotide is altered, it doesn't have this OH group, right? It doesn't have it. It's missing. So whenever one of these dideoxynucleotides adds, no other nucleotides can add after it. Okay, and that's important because it terminates this particular DNA synthesis is terminated. Now, there's lots of DNA in there, so there's all kinds of DNA strands that are being amplified at the same time. And because the dideoxynucleotides add randomly, what you'll end up with is you'll have lots of DNA at the end, but they're all going to be of different sizes because depending on where the dideoxynucleotide added, if it adds early, then the fragment of DNA is going to be small. But if it adds late, then it will be large. Okay? So in that way, you can determine where, like at which position, the dideoxynucleotide added. Okay? And the way you can do that is you run it through a gel, just like you would with regular DNA in an agarose gel. You run it through and you can see the size of the DNA fragments. But in this case, the sizes of the DNA fragments correspond to the position at which the dideoxynucleotide added. I see how these little stars are representing the dideoxynucleotide. So these guys, because the OH group has been removed, it terminates the reaction, so the DNA can't amplify anymore. That strand of DNA, right? But they also add a fluorescing molecule in its place. In the place of the OH, they add this little flag that literally glows under UV light. And when it runs through a machine, a laser can detect the color that it glows. Every dideoxynucleotide, it glows a specific color, depending on whether or not it's an adenine or a thymine or a cytosine or a guanine. So the laser can read these colors and it can tell which dideoxynucleotide added at which position, and you end up with an entirely sequenced piece of DNA. It's really a marvel of science. So now we're in 12.3, whole genome methods, pharmaceutical applications. So The science of genomics allows researchers to study organisms on a holistic level. So we really have got a lot of information now, way more than probably Watson and Crick ever dreamed of. Transcriptomics and proteomics can compare gene expression patterns. They can help figure out how to respond to pathogens. You have immune cells and white blood cells, and how those genes are turned on or turned off in relation to, bacterial or viral infection. It's just an amazing field of science that I couldn't possibly go into all the applications here. These things complement each other, so you can look at genomics and proteomics and transcriptomics and all of these omics, and it ends up being basically a new science called metagenomics. And you can look at how gene expression affects protein production, how having the genes at all, which genes express what and under what circumstances, and how everything affects everything else. It's just awesome. And then recombinant DNA, this gets a lot of controversy and stuff like that, but it makes GMOs, genetically modified foods, that are really honestly feeding the world right now. And also they're used to make pharmaceuticals. RNA interference is a relatively new science where they are learning how to physically enter into these transcripts and turn them off. It's awesome. They can actually silence genes that when they express, they have deleterious effects. They are now really working hard to be able to go in and silence these genes. And I'm talking about stuff that turns on cancer cells, and it's really awesome where the science is going these days. You can see I'm excited. I know, I'm geeking out. I'm sorry. This is a diagram that's illustrating the process of RNA interference in a eukaryotic cell, and it is silencing a gene that has to do with pathogenesis of some kind of a disease. And I don't expect you guys to know the ins and outs of RNA interference. It's just know that it is a science and that this is the way it works. And basically what they do is they make a very small modification in the mRNA such that the mRNA can't be translated into the correct protein. So, if you change the protein's shape, you can make it non-functional, right? So essentially that is what RNAi is all about. And then, of course, the last thing I want to talk about briefly is gene therapy. It's really showing great promise for the treatment of genetic diseases. If you know what genes cause what diseases, that's step one at being able to turn the expression of those genes off or to mutate them or otherwise. But, it is a budding science. It hasn't been that long that we even were able to map the genome of humans. So it's still in its infancy. And it runs into issues too, because you have a lot of federal and local regulation that slow down the process of gene therapy. There's ethical issues, but it's the wave of the future, and perhaps just maybe we can cure cancer and diabetes and heart disease. I remain optimistic. Well, I guess we'll have to see. Maybe before I die, gene therapy will cure cancer. So that is going to do it for lesson 10. This was the last lesson as part of my Fundamentals of Microbiology course. I want to thank you guys all for watching this and my other videos. As always, don't forget to do the reading. Check the description below for more videos related to these topics, and leave your questions for me in the comments section below. Bye!